Good afternoon to everyone once again. On behalf of the organizing committee, I, Suhas Kaushik, faculty in the Department of Mechanical and Industrial Engineering, welcome you all to the session of another session of the International Conference on Recent Advances in Fluid Mechanics, ICRAFM. As we all know, an international conference is an opportunity to listen to the novel ideas of bright minds of the world. And we also know that we get a bonus too in the form of a keynote speaker who share their experience and shed light on the future of the research area. Today, we have with us a highly decorated researcher and eminent personality in the area of computational fluid dynamics. As our keynote speaker, Dr. Suman Chakraborty. A warm welcome to you, sir.
Yeah, good afternoon, and it's a really great honor to be a part of this conference where there are several distinguished speakers and participants, and I'm glad that we are here. Of course, physical mode would have been adding another flavor, but nevertheless, it gives us an opportunity to discuss and interact on various aspects of research related to the focus or the focal theme of this conference.
Thank you, sir. And it is my duty to introduce you to our audience before we move on towards your presentation. We know it's a uh, very brief profile that I must get to because your achievements are so huge. So getting to it again, to all the audience, I would like to introduce our keynote speaker. Dr. Chakraborty is currently serving as a professor in the Department of Mechanical Engineering at IIT Kharagpur. He has completed his undergraduate studies in mechanical engineering from Jadavpur University in '96, master's degree from Indian Institute of Science. He received his PhD in 2002 and was bestowed with the Institute level Best Thesis Award, as well as Best International CFD Thesis Award for his work on solid liquid phase transition during thermal processing of materials. Further, throughout his career, he has had many notable achievements. Some notable discoveries emerging from his research would include surface roughness aided slippery flow, massively amplified ionic pumping in highly confined water, and the list goes on. Dr. Chakraborty is also known for developing disruptive medical diagnostic technologies for the underserved. His innovation on a new piecewise isothermal nucleic acid test, COVI-RAP, has emerged as the first of its kind, highly accurate molecular diagnostic technology for infectious disease detection. This generic platform, applicable for several diseases beyond COVID-19, has been certified by regulatory authorities and commercialized for global dissemination. Needless to say, at this point, Dr. Chakraborty has been decorated with many awards and fellowships for his significant contributions in the field of research and academics. To name a few, Sir J.C. Bose National Fellow by the Ministry of Science and Technology, Government of India, Fellow of the Royal Society of Chemistry, United Kingdom, recipient of the National Academy of Sciences India Reliance Industries Platinum Jubilee Award for Application-Oriented Research, recipient of Rajiv Goyal Prizes in the area of Physical Sciences 2021, and again, the list goes on. Further, speaking of certain statistics, Dr. Chakraborty has published more than 850 research articles with nearly 13,000 citations. His H index currently being at 59 and I-10 index at 354. He has 11 patents to his name, and he also has 11 technological transfers to companies to his credit. And today he has joined us to present on the interesting topic, as you see on the screen, material engineering interfaces microfluidics towards probing cancer metastasis on a chip. Sir, we are indeed honored to have you amongst us. Over to you, sir. Request you to kindly address the session.
Thank you very much for this very kind introduction. And as I mentioned, that this is a great platform of interacting with various specialists in different aspects of fluid mechanics, transport phenomena, having different applications. Now, that of course creates a diversity, but at the same time, we need to recognize that everybody may not be that much familiar in the particular field in which we are deliberating on. So this is an area where we are discussing about the convergence of fluid mechanics, materials engineering, and overall microfluidics to understand cancer metastasis on a chip. Now, just to give it a perspective, the cancer metastasis is a physiological puzzle, and to give you a rationale of why is this a physiological puzzle, imagine the human circulatory system. We have large arteries, large veins, which are I would say macro channels in human body. You have small arteries, small veins, arterioles, venules, and tiny capillaries which connect the arterioles and the venules. These tiny capillaries are human body microchannels. Now, all what it means is that when we are considering fluid dynamics or blood flow dynamics in particular, in human body circulatory passages, you have flow for over a wide range of Reynolds number, and also in blood vessels having different deformabilities, different geometrical attributes, and most importantly, different functional connects. Now, how does this relate to tumor metastasis or cancer metastasis? All of us know that the most lethal stage of cancer comes when a cancer cell from its primary site moves along the bloodstream and reaches a distant site which is far away from its original location and creates a new cancerous trait at that location. That is how cancer can spread all around the body, and if it does so, it gets extremely difficult to treat it, which otherwise would have been much more convenient had it been confined locally. So you see a diagram here where you have a primary tumor and you have some transformed cells. So these transformed cells typically would have a transformation from epithelial to mesenchymal cells, and these transformed cells are there in the tumor microenvironment and they are shed into the bloodstream. Once they are shed into the bloodstream, they will move around, and while moving around, of course, one of the major pathways that they have to cross, and that is the most difficult barrier, is the microcapillaries. So in the process of this cell migration, there are several events. First, new blood vessels are formed, which is known as angiogenesis. Then the cancer cells adhere to and invade the basement of the membranes, Then they pass through extracellular matrix. You can see that they are passing through the extracellular matrix. They have intravasation, that means they enter into the vascular pathway. They interact with the host lymphoid cells and form tumor cell embolus. They again adhere to the basement of the membranes at certain other locations. Now the extravasate, that means they get out of this main blood vessel and have a metastatic deposit, and by creation of new blood vessels, they reach a secondary tumor site and start growing. It means that the microvascular pathway, the blood vessels, the microchannels in human body are extremely critical elements to support this movement of the cancer cells and create its metastasis. Now all that is known and of course qualitatively well understood over the years. The challenge is that within the human body, how can you make fluid mechanics experiments? That means how can you get the details of how these cancer cells are moving from one location to another. Therefore, it is quite interesting to develop artificial microfluidic devices. That means instead of having the study within the human body, we make certain small channels in which we study the dynamics of cancer cells. Or to begin with the discussion, any particular cell, be it cancer, be it normal. Now these kind of studies, which we call as in vitro physiology, these are extremely critical for several other reasons, not just for studying the cancer cell migration, but also doing research in the drug discovery. For a long period of time, the classical way of doing drug discovery research is the drug development and preclinical trials in the lab, which include small animal trials, maybe large animal trials in certain cases. So altogether, this part of the advancement will take more than 6 years on an average, sometimes more. And then maybe for another 7 years, trials on humans at different phases. That means that starting from the discovery to use for the benefit of humans, it may cover a cycle of about 14 years. On the top of that, when we are doing lots of trials with animals or humans, there are inevitable ethical concerns. Nowadays, these are extremely serious and dealt with in a manner where lots of protocols and guidelines and statutory measures are taken. On the top of that, several animal experiments give results which are very, very different from the results obtained from the human studies. This gap in understanding or this gap in the animal and human models, it reflects the lack of certain physical similarity between maybe a traditional in vivo cell culture system in animals, in vitro cell culture system that we get in the lab, traditional lab, and in vivo systems inside the human body. Now with microfluidics and tissue engineering, there has been a whole lot of advancement in creating 3D in vivo conditions, or I would say in vivo like conditions, not exactly what happens in the human body, but very much with similarity. And one can do a whole lot of detailed control studies in that artificially bioengineered system, which is not possible to do inside a human body, or in fact also not inside an animal body. Therefore, there is a significant thrust in developing such artificially engineered bioanalytical tools for maybe first cell on a chip and then create a connective system to understand the entire cell on a chip from a systems perspective. This is an early study done by my former PhD student Dr. Tamal Das, who is currently a faculty member at TIFR Hyderabad, that if you have a cell which is sitting on a microfluidic channel, what is the role of the fluid flow in altering the dynamics of the cell? So towards that, we developed a technology which is known as traction force microscopy on a chip. What is this traction force microscopy? You have a cell which is adhering on a microfluidic substrate, and there is a fluid flow which is administered around the cell. The cell is standing on the microfluidic channel. In this case, it is a PDMS, that is a soft polymer, and it can be made ultra soft so that it is very flexible, and the interaction of the cell with a flexible blood vessel wall can be mimicked to an extent, not completely, through this arrangement. So just like as humans we use our legs to stand on a surface, the cells also have their own legs, and these legs are known as focal adhesion points. If we now have a fluid flow on the cell, then what happens is that it is very similar to, say, as humans we are standing on the ground, but somebody is giving us a kick at our back. We will feel destabilized on our feet first. The same thing happens here. The cell gets destabilized at their feet, which are known as focal adhesion points at the basal surface. Once the cell gets destabilized on its feet, the entire body of the cell starts getting a feel that there is a mechanical stress. But how does that message propagate throughout the body of the cell and reach the most distant part of the cell membrane, which is called as apical part of the cell membrane? It is by a mechanism called as mechanotransduction, which is essentially a mechanism by which mechanical signals propagate within biological systems. So once the apical cell membrane feels that there is such a mechanical disturbance, it responds to that. How? The cell membrane has a layer of lipid, which is basically like a kind of soft material, very soft liquid like material, but inside that you have some solid, more solid like entities which are called as lipid rafts. So when the cell membrane understands that there is a stress, the lipid rafts try to adjust to that by escaping from cell membrane to inside the cell. In that way, the cell membrane which has lipid raft, now there will be less lipid raft in the cell membrane because more part of the lipid raft has escaped from the cell membrane to inside the cell. All what it means is that the cell membrane becomes more fluidic, and because it becomes more fluidic, it becomes more malleable and it can have a unique shape adaptation to survive the stressful condition. And in fact, if the cell is within a microchannel, that is what it does. It adapts its shape because of the extreme confinement to make it survive under such stressful conditions. Now, how easily it can have a shape adaptation? It all depends on how much fluidic the cell membrane is, and we also developed certain techniques to measure the cell membrane fluidity, which implicitly gives an indicator of how quickly the cell membrane responds to the stress acting on it. And accordingly, the cell deforms, and we can, in the process of this traction force microscopy on a chip, we can draw an outline, and by the displacement characteristics of embedded beads within the microfluidic channel, we can reconstruct a force by an inverse Fourier transform, which could be responsible for such displacement. And we can draw like a stress map around the cell with some zones of high stress concentration like this reddish zone, and the zones of low stress concentration like the bluish zone. Now this is something that as a research we did say 10 years back. Now 10 years down the line, what we progressively realized that, well, this is a good sort of insight on how a cell interacts with a standalone microchannel, but human body microfluidics is not just a standalone microchannel. If you see the blood vessels have very complex network structure, and because they have a very complex network structure, what happens is that the blood vessels, they create a very unique type of resistance as any biological cell invades through that network. And this network is much more complicated than a piping network that we study in fluid mechanics in traditional engineering. On the top right view graph of the slide, you see the piping system in a very large engineering plant. As engineers, we think that these are extremely complex. Indeed, these are complex. But in the bottom right view graph, you see human body piping network or human body microchannel network as blood vessels are dynamically created as the cancer metastasis takes place. This is angiogenesis or dynamic creation of a complex network of blood vessels. We can realize that the complexity of a true human system is unparalleled, and therefore it is perhaps not at all possible to exactly mimic that in an engineered microfluidic environment. But having said that, there are logical steps that one could undertake to move way forward to understand how the tumor cells and maybe the blood cells move across the complex network of blood vessels in the human body. If you see the blood as an example, it carries with it some additional elements of complexity that a traditional Newtonian fluid like water will not have. Summarily, blood has a watery component which is plasma, and the cellular components which are the red blood cells, white blood cells and platelets. Out of that, the red blood cells are extremely critical for influencing the fluid dynamics of blood. One of the very common sense reasons is 85% of cells in human body are red blood cells, and 45% roughly of the blood is made of red blood cells. Now this volume fraction of the red blood cells in blood is known as hematocrit. So the blood flow in human body is extremely complex, not just because of this non-Newtonian nature of this blood flow, but also for the fact that under certain conditions, these blood cells, these red blood cells rather, form certain layers where the wall adjacent layer is depleted from blood cells and the far away from the wall is rich in the blood cells. This leads to a very, very peculiar phenomenon which is known as Farhius-Lindquist effect, which was brought out by these two famous physicians in 1931. But only of late, maybe last 5 to 10 years, people have been in a position to explain this to some scientific accuracy and foundation. So summarily, if you have these blood cells in a highly confined passage, the blood cells will be subjected to two major types of forces, one towards the center line of the channel and another away from the center line. So there is a shear gradient because of the pressure driven velocity profile, which will drive the cell towards the wall. But there is a wall effect, and because of the deformability of the red blood cells, there is a wall induced lift force which will take it away from the wall. So when these two forces are in equilibrium, the red blood cell gets located at a location which is away from the wall. That means the near wall region gets depleted of the blood cells, and the near wall region is the home ground of creating high viscous resistance. So all what it means is that what you have is an effect where there is tremendous sort of pull away from the wall, and the blood therefore appears to be less viscous. So up to this we have discussed about the blood flow. I want to take a bit of 5 minutes of break, and I will come back and continue with my talk. Just 5. Okay, so let me move on. So now I will continue with what I was talking about, that therefore the viscosity, the effective viscosity of blood as it is moving through a microfluidic confinement is not as straightforward as one would imagine as the diameter of the blood vessel reduces to an extent that it becomes several microns, it becomes almost close to the average dimension of a red blood cell, which is around 8 micron end-to-end biconcave disc. And the deformation is so severe and the deformation induced lift at the wall is so severe that the reduction in effective viscosity is very strong. So we have seen that the red blood cells deform and they influence the dynamics of blood flow. What we have so far not seen, that do the microchannels deform? Of course, in human body they do, but do the microchannels on which we are doing our in vitro studies, do they deform? Here we have presented a scenario in which a very frugal manufacturing process is established to fabricate deformable microchannels. These microchannels are made of a material called as gelatin, which is a derivative of collagen, a protein abundantly found in the human tissues. These proteins are such that any material which is derived from such proteins, such as collagen, will have excellent biocompatibility, and the biocompatibility here is much more than PDMS. They are deformable to an extent that they can be designed in composition to mimic certain tissues of various organs in the human body. And how do you make such microchannels? Very easy. You take a petri dish, you use a lumbar puncture needle, which is commonly used in the medical context and medical procedures. Typical diameters between say 240 to 480 micron, and you pour this gel material around that. Once it solidifies, you take away that needle, and in that way a hollow cylinder of that much dimension is created. Now we had our first experiment with such a channel by filling up a syringe with red blood cells, and then via a flexible polymer tube, a Teflon tubing, we passed the red blood cell solution through this channel. We tried to track how the red blood cells have their collective motion by a method called as particle tracking velocimetry, and we of course use the traditional PIV, the particle image velocimetry, to evaluate the velocity field in the channel, and the wall deformation was measured from microscopy. Now what we could find out, and it was a very interesting observation, that the red blood cells typically they will have tumbling motion, but they can change the tumbling motion to tank treading motion, and this is known as tumbling to tank treading transition. It is basically a rotational motion to I would say that the movement where the rotationality is very less. the transition from tumbling to tank treading is known to be one of the very critical hallmarks of the red blood cell dynamics in a microchannel. While it was understood that this transition happens in the human body microvasculature, it was also understood that how a microchannel would influence that, but all the previous experiments were based on rigid microchannels. This was the first time that we did experiments systematically with deformable microchannels and saw that how the deformation is influencing this transition. But then the real human body system is not a single deformable channel. It's a network of deformable channels, like almost like the branches of a tree. So what we try to do, we try to develop certain micro fabrication procedures which are frugal and at the same time replicative of what happens in a branched structure of flow. So what we did is we used, or rather we developed a technology which is known as leaf inspired soft lithography. So what we did is we took a leaf, pasted it inside the petri dish, covered that with PDMS, peeled off the PDMS to get a negative replica, and it was covered with PDMS after sigma coat treatment, and then it is peeled off, so you get basically a replica of the same leaf, but not the living system, PDMS replica, and then you can just connect that with the inner and the outer, the inlet and the outlet channels. So in that way, on a glass slide, we could have this PDMS channel in this way, but what we realized is that in this way, whatever channels we get, the inner lumen of these channels is basically having a shape which is not the circular wall of a blood vessel. So what we did is we tried to develop an alternative strategy with a similar network structure. So what is that alternative strategy? We spin coated the PDMS on a clean glass slide. We patterned agarose gel wires pneumatically taken out of a Tygon tubing on a cured PDMS gel side. So these are basically gel wires made of gel. Then we poured PDMS on the top of that agarose gel wire pattern, and then we flushed out the melted agarose with hot water using a syringe, and that means now you have hollow microchannel, and then we can connect it with the reservoirs. So you can see the photograph of these kinds of channels, and you see the lumen now, while the previous structure had a non-circular lumen, now it is having a circular lumen. Now comes a bit of mathematical modeling. Well, the circular lumen flexible microchannels are made, but what happens during the cancer metastasis? In that case, these blood vessels are not static but dynamic, and the metastasis is something that takes place. So how does it happen? You see a healthy stable vessel, then let us say that there is a primary tumor, and the tumor has certain chemicals around it which is called as growth factor. So the growth factor is high around the tumor and is low at the blood vessel which is away from the tumor. So a concentration gradient of this growth factor is created, and in that way, because of this concentration gradient, there is a migration of the endothelial cells from the wall of the blood vessel. They proliferate, and they actually move in the direction of this highly rich concentration of growth factors. and they cannot just move like that, so they will dynamically create blood vessels and sprout rather blood vessels, and then that blood vessels they will form their own network or branches and converge to the tumor site. In that way, new blood vessels are formed. So what we did is we tried to make a very simplified physics-based model where we had the parent blood vessel, the VEGF source, which is that concentrated location in the tumor, and this is the extracellular matrix, which is the blue colored region, and there is an exchange of fluid, and so what we did is for the extracellular matrix, which is this passage, we used a porous medium model, and for the channels and the connectors we used the traditional fluid mechanics model. So we studied two things. One is the concentration of the growth factor and the concentration of the endothelial cell, which is in this view graph, the variable n. So we developed a mathematical model which would relate that n with the fluid pressure and the concentration of the growth factor. So in that way, we came up with a distribution of the endothelial cells, and therefore by reconstructing that distribution, we could get a physical picture of how the tumor metastasis could take place. Now, once we realize that, we try to move further forward and try to see, well, these endothelial cells, they are very, very important, and these endothelial cells are there at the walls of the blood vessel. Now, how they are interacting with the tumor cells? This is the whole crux of the metastasis problem. So we have the circulatory tumor cells, which we studied their dynamics as they are moving along with the red blood cells and they are interacting with the red blood cells. We tried to get a complete imaging map of how these endothelial cells are there, and now we could characterize the cell-free layer of the red blood cells. We could locate the nucleus of the cells. You can see this blue colored dye, and cell-cell tight junction, which are the green colored materials. So in that way, we could realize that the endothelial cell is indeed sort of there at the wall, and the other cells, including the circulatory tumor cells, are migrating. Now, the question is, when the cells are migrating and they migrate in a certain way, they create a colony and they create a new cancerous trait. There is no doubt about it. Now, can we use the same physics to actually have a targeted drug delivery mechanism where the drug will be directed to the same location where the cancer cells are migrating? So this is a study that we very recently reported and performed, that we tried to develop a magnetic nanoparticle assisted targeted therapy protocol, where the first part of the study which we had completed is, instead of the drug, we just had these nanoparticles, magnetic nanoparticles, and these nanoparticles are moving with the bloodstream. Now, if we apply a transverse magnetic field, the nanoparticles will go towards the wall, but they may not be sufficient, or they may not be in a position to have sufficient strength to actually make a proliferation and get directed to the tumor sites. If they get directed, the good thing is that they may carry the drug and have a very targeted delivery of drug at those locations. So how does the deformation of this microfluidic channel can help in this nanoparticle assisted transport? To understand that, we use a hydrogel-based microfluidic system. We fabricated that and studied the nanoparticle assisted targeted drug delivery. We found out, how the bulging of the walls, the deformation of the walls, is actually facilitating the transport of these, I would say, nanoparticles, because if they are coming down more towards the fluid, they are engulfing the nanoparticles, and the nanoparticles in that way, they are in a position to proliferate more into this extracellular matrix. So what you are seeing here is that you have a wall, the wall gets deformed, and it gets deformed to an extent that the nanoparticles will penetrate with the aid of the deformation and reach the tumor site. And there can be a controlled delivery where there will be a harmonization of how the walls of the blood vessel deform and how the nanoparticle gets transported. In many of the experiments which were previously done with rigid channels, the nanoparticle aided drug delivery did perform excellently in the artificial system, but did not work in practice because the physical model did not take into account the interplay between the wall deformation and the movement of the nanoparticles. Now, of course, those have been very early studies, and we are yet in a position to actually see the complexity of the drug targeting, the tumor metastasis, all combined together in a human body network. So in a real human body, you of course have these blood vessels, which we conceptually we are showing by green colored lines, but they are connected by organs. And once you have a drug targeted for a particular disease site, inevitably inside the human body, the other major organs get affected, which we know as side effect of the drug. And this is because of the transport across this connective network. So this on a small chip, if it is replicated, that is known as human body on a chip. That concept again has been known for about a decade, but till now we do not have a biomimetic human body on a chip. That means a human body on a chip model which to a large extent mimics the structure and the functional aspects of the connective system inside the human body. So that is where the direction of further research is moving. That is developing biomimetic human body like microfluidic networks, studying the dynamics of cells, drugs, etc. under electric magnetic fields or even under pressure driven flow. And this may lead to a complete bioengineered biomimetic platform, which will of course never be able to replicate what it happens in the human body, but will take us sufficiently closer to what could have happened in human body in terms of cancer metastasis, drug delivery, and treatment of extreme critical diseases. I'd like to thank all my co-researchers, colleagues, and students who had worked on these topics, which are extremely challenging and complex. We are still at the infancy in terms of understanding even the most essentials of these topics, but I think that we have developed a foundation based on which we can move forward. So thank you very much for listening patiently to this discussion or talk, and I would be happy to take maybe a couple of questions before we can perhaps conclude this session.
Thank you, sir, for that informative session. Regarding the Q&A, there is one question from Dr. Zubair. Sir, are you able to see the question in the Q&A section?
Let me open it. I'm sure I will be able to see it. So it says that no new questions. I do not know. I mean, maybe it is not coming to me. I
Okay,
have
sir.
opened this one.
There is one tab called published. Is there a tab called published questions?
Yes. I see 2 published questions.
Out of that, the second one from Mohammed Zubair.
Yes. How is the forces acting at the center line and those developed due to shear affect the blood flow? Yeah. So this is a very important question. So first of all, there are 2 critical forces that we are talking about here. One is that had there been no deformability of the cells, the cells would be subject, or say instead of cells, if you could consider microparticles, they will be subjected to a force due to the shear gradient, and that shear gradient is because of the Poiseuille flow or pressure driven flow parabolic velocity profile, and that will drive the that will create a driving force towards the wall. What happens is because of the microfluidic confinement and the deformability of the cells combined together, there is a wall induced lift force, and wherever this interplay is strong, the lift force away from the wall is also stronger, and then it will keep or levitate the cell at an in between position between the center line and the wall. More the wall induced lift force, more it will go away from the wall, and it can be controlled by the deformability of the wall and of course the deformable characteristics of the cell.
Right. Thank you, sir. I think Dr. Zubair, that sir has answered your question. If any other participants have any queries, please post it here.
Okay, I think that it was anyway a bit of a heavy topic for because not that it is sort of extremely out of the way, but normally these kinds of topics are not covered by fluid mechanics researchers. So in that way, it will be different. But if not now, I will be very happy in welcoming discussions and questions on these topics maybe later on. But for the time being, I think that I must thank you all for giving me this opportunity of delivering this talk, and thank you very much once again, and possibly goodbye for the time being.
Thank you, sir. Thank you very much.